Hello, my dear students. Very happy to meet you all again through this video. In this presentation, I'm going to discuss with you about cell culture. This is the last unit in your syllabus. So now let us discuss about cell culture. So the topics covered under this title is how to grow the animal cells in culture. Okay. So in the intro part, I will discuss with you about what do you mean by cell culture technique. And next we will move on to the general procedure for cell culture. And next comes what do you mean by the different types of cell culture, that is primary cell culture, established cell culture and transformed cell culture. And at last we wind up with the applications of cell culture, especially in pharmaceutical industry and research. Cell culture is nothing but previously we learned about the cultivation of microbes. Okay, how to cultivate the microbes. Here we are going to cultivate the cells in a controlled environment, in an artificial environment. That artificial environment is controlled by us. Okay, so controlling in the sense we can able to maintain the required things which promotes the cell, and also we can able to control the growth of cells to the extent how much we need. Okay, so here the cells are isolated from the living beings, either the plants or the animals or the human beings. Okay, so these cells are isolated by a process. After isolation or separation, they are allowed to grow in a controlled artificial environment. Okay, so the cells can be maintained in vitro condition. So in vitro condition is nothing but outside the living environment. Say for example, if we are isolating something from animal's body, okay, so we are making the cells to be alive, or making the cells to multiply out of that animal body. Okay, if I am isolating any part or any cell from cat, or any cells are isolated from dog, or any cells are isolated from monkey, or even we are isolating the tissues from plants, outside the plant, outside the body of animal, we are trying to promote the growth of those cells by providing all the supporting conditions. Okay, but again, this process, it is quite simple compared to organ and tissue culture. Okay, the cells can be maintained in vitro outside of their original body. So here the process is quite simple if it compared to organ and tissue culture. In a cell culture technique, just we are removing the cells and it is grown in a favorable environment. For the purpose of animal cell culture, the cells are taken from the organs of the experimental animal, and these cells may be removed directly or by means of mechanical method or by using some enzymatic action. Okay, so direct removal, mechanical method of removal, or enzymatic method of removal. So either of by these means we can able to remove the cells from the organs of experimental animal. So the cells which was obtained, it can be made as a cell line, or we can call it as a cell strain. Okay, so which is nothing but again we are going deep into the cells, okay, to the subunits. Say for example, the cells which are used to culture are fibroblast, lymphocytes, cells from cardiac and skeletal tissues, and cells from liver, breast, skin, kidney, and many different types of tumor cells. So using this, we can able to maintain the cell line, or we can call it as a cell strain. That is, it is preserved for future purpose, or it is preserved for further processing. So now comes growth. So how to grow? So in previous slides, I discussed about how we are removing the cells from the living beings. So now how to promote the growth of animal cells? Again by means of using culture. And culture, in case of culture media, what we used for growing the bacteria and fungi, that is quite different. So here typically it requires more nutrients, since it is an animal cell or a human being or a plant. it is a living cell. Okay, so it is very difficult to culture the living cells, that is animal cells, compared to microbes. Compared to microbes, the growth of animal cells under in vitro condition is very difficult. So here it requires more nutrients compared to microbes. And also the grow can occur only it attaches to a specially coated surfaces. Okay, the growth can takes place only if it attaches to specially coated surfaces. Again, even though many difficulties are there to grow an animal cell, including both undifferentiated or it may be a differentiated cell, it can be cultured successfully by means of using in vitro laboratory conditions. So, what are all the requirements for these cultivation of animal cells that we will see in detail? Here I have given one media as the example for promoting the growth of animal cells or mammalian cells. Serum containing media, which is Eagle's media. Okay, here it contains essential amino acids, which includes histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Along with that, it also contains cysteine, glutamine, and tyrosine. So here the concentration is also mentioned 10 to the power of minus 4 to 10 to the power of minus 5 molality. And it includes major vitamins like folic acid, nicotinamide, pantothenate, choline, pyridoxal, and thiamine. Apart from that, it also includes inositol and riboflavin. And major salts present in this Eagle's media are sodium, potassium, calcium, magnesium, chloride, phosphate, and hydro bicarbonate ions. Okay, and it includes glucose and dialyzed serum. So this is an example for serum containing media to promote the growth of animal cells. Next comes serum free medium. Okay, so here it is coming under defined culture media. So while discussing about the different types of culture media, there are two types: defined culture media and undefined culture media. The previous one, serum containing, it is coming under undefined culture media. So here it is a serum free medium which comes under the category of defined culture media. It contains amino acids, and almost the composition of amino acids is similar to the Eagle's media. Along with that, it includes alanine and asparagine, and the same vitamin, salts, and glucose, same as that of Eagle's media. Okay. Apart from that, it includes fatty acids, nitrogen compounds, carbon source, trace elements, and some hormones and growth factors. And these are very much helpful to promote the growth of animal cells under artificial environment. Next comes the different types of animal cell culture. Okay, so we can categorize this animal cell culture based on two criteria. Okay, so first one, based on the number of cell division. Based on number of cell division, this animal cell culture is categorized into two types: primary cell culture and secondary cell culture. Okay, so again, this primary cell culture, based on the nature of cells, we can categorize them as adherent cells, suspension cells, and another category, confluent culture. Okay, confluent culture. Here we can discuss about the necessity of the subculturing technique. Okay, and second category, secondary cell culture and cell line. So here the cell lines, what we are deriving from secondary cell culture, it could able to undergo finite or infinite cell division. Again, it's a general term. Finite means with the prescribed number of cell divisions. Say for example, 5 number of cell divisions takes place, 10 number of cell divisions takes place. Like that, we can able to represent or denote how many number of cell divisions is possible with the available cell culture. Whereas infinite cell division, it exclusively covers the tumor cells. Okay, here we can't able to tell how many number of cell divisions will occur. Okay, so it is divided as finite cell division and infinite cell division. This is different types based on the number of cell division. Next comes the type of cell culture based on the lifespan. Okay, so as I told you, whether the cell lines can able to divide for the particular number, that we can call it as finite cell lines, and another one is continuous cell line. Apart from that, the cell lines what we have derived, it is categorized under common cell lines. Okay, so first we will discuss about the previous one. Okay, primary cell culture. So primary cell culture, which is nothing but primary and secondary, the term is common. Primary is one which is having direct contact with the source. Secondary, it is derived from the another source. It is not having indirect touch with the resource or direct touch with the raw material or direct touch with the things that is concerned. Okay, so primary cell culture, these are all the cell culture obtained directly, obtained straight from the animal tissues or obtained directly from the host tissues. Okay, so here the cells are disassociated from the parental tissue. So these cells, after disassociation, they are allowed to grow on a suitable container. Okay, so initially we are separating the cells from the parental cells, or we can call it as the host cell, and it is allowed to grow on a suitable container. So this type of culture is called as primary cell culture, and mostly it comprises heterogeneous type of cells. Mostly it comprises heterogeneous type of cells, and these cells can able to divide only for a limited time period. Okay, but these cells are very much similar to their parents because they are directly derived from the parents, so they have utmost similarity with their parents. Here I have given one media as the example for promoting the growth of animal cells or mammalian cells. Serum containing media, which is Eagle's media. Okay, here it contains essential amino acids, which includes histidine, isoleucine, leucine, lysine, methionine, phenylalanine, adherent cells. So the name itself indicates they has to be attached to the surface. Okay, so these cells are anchorage dependent. Okay, they grow or they promote their growth as a monolayer. Okay, so just imagine that over the surface of water some layer has been present. So these cells need a solid support. Okay, or a semi-solid substrate is required for the proliferation. Proliferation is nothing but growth. So adherent cells are nothing but they are anchorage dependent. That is, they need a strong solid support to promote their growth. And again, if any solid support is provided to them, then they can able to grow as a monolayer. Monolayer in the sense only once they can able to grow over the solid surface. Above which another layer it cannot able to develop. Okay, so it needs a solid or a semi-solid substrate for promoting their proliferation. These adherent cells generally they get attached to the culture vessel. Okay, so to which vessel, to which container we are cultivating, we are introducing them, it's supposed to adhere to the culture vessel with the help of their extracellular matrix. Okay, so extracellular matrix is nothing but this is a type of substance that is derived from the tissues of organs what we have isolated. Okay, and these are immobile and they get embedded with the help of a network of connective tissues. Okay, they develop an extracellular matrix. With the help of this matrix they get attached to the culture vessel. Okay, so from where this extracellular matrix is obtained, the tissues what we have inoculated, that tissues produce this extracellular matrix by its own, but they are immobile in nature, but they can able to provide a network of connective tissue. These includes, okay, so generally it covers different types of cells. For example, fibroblast and epithelial cells. fibroblast and epithelial cells. See, if you remember in your HAP, you might be learned about clotting, okay, how the blood clotting takes place. So how the blood clotting takes place, even in your practical class, you have done with the help of capillary tube method. So while breaking down the capillary tube, you supposed to observe the development of fibrin clot, blood clot has been developed, which is nothing but a small mesh-like network is produced over the bloodstream or over the wound or over the broken area. The same way here, the cultured cells are grown as a adherent monolayer. Okay, so when the bottom of culture vessel is covered with a continuous layer of cells, usually it is about one cell thickness. Okay, only one cell, it is present as a layer, but if you measure the thickness, it is around only one cell. Like that, we can tell it is like a horizontal construction. One cell cannot grow over the another like a pile of coins. If we are having 10 number of coins, so 10 number of coins, if you are keeping one over the other, that we can call it as the pile of coins. Whereas if you are arranging the 10 coins, one by the side of another, one by the side of another, like a horizontal line, imagine like that, the same way the adherent layers forms. So this we can call it as a monolayer culture. Majority of continuous cell lines, they grow in the form of monolayers. Okay, so being present in the form of single layers, they can be transferred directly to a cover slip. Okay, you can remove one particular cell easily and it will be directly transferred to the cover slip, so that you can able to examine under the microscope and you can study the property of those cells. Okay, so this is about adherent cells. So under primary cells, there are two types, adherent cells and suspension type of cells. Next comes suspension cells. Okay. So here the suspension cells, so the suspension cells do not attach to the surface of the cell culture. Okay. So here the first line, actually it is a adherent type. Fibroblast and epithelial cells, it belongs to adherent type. By mistake, the same line gets repeated here. You please delete the first line. So first line, it is coming only under adherent cells. Okay. So here the cell suspension, it do not attach to the surface of culture vessel. Okay, so the name itself indicates suspension, it is present in the liquid form, it gets suspended in a vehicle. Okay, so here these cells are anchorage independent. Okay, or we can refer them as non-adherent cells. It can able to grow floating in the culture media, it can able to grow as a floating in the culture media. And here the examples comes hepatopoietic stem cells, which is derived from blood, spleen and bone marrow, and all tumor cells can be grown in the form of suspension cells. Compared to adherent cells, the suspension cells can able to grow much faster, which do not require frequent replacement of medium. Okay, so here compared to adherent cells, here it is very easy to maintain. And another difference, adherent cells are heterogeneous type, here it is homogeneous type, and so enzyme treatment is not required for cell dissociation. That is, for cell separation, enzymatic treatment is not needed. And similarly, these cultures have a short lag period. This is a comparison between adherent cells and the suspension cells. Here I have given one media as the example for promoting the growth of animal cells or mammalian cells. Serum containing media, which is Eagle's media. Okay, here it contains essential amino acids, which includes histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Along with that, it also contains cysteine, glutamine and tyrosine. So here the concentration is also mentioned, 10 to the power of minus 4 to 10 to the power of minus 5 molality. And it includes major vitamins like folic acid, nicotinamide, pantothenate, choline, pyridoxal and thiamine. Apart from that, it also includes inositol and riboflavin. And major salts present in this Eagles media are sodium, potassium, calcium, magnesium, chloride, phosphate, and hydro bicarbonate ions. Okay, and it includes glucose and dialyzed serum. So this is an example for serum containing media to promote the growth of animal cells. So because of subculturing, we can able to maintain the viability of the cell, or we can call it as liveliness of the cell. A passage number. So here comes the passage number. It refers how many times a cell line has been subcultured. Okay. See, initially I am isolating liver cell. Okay. So one organ I have isolated, liver. So from the liver I have isolated a cell that we can call it as a liver cell. So this liver cell which is isolated directly from the liver, this is called as primary cell culture. So this primary cell culture, after 2 days or after 3 days, if it becomes overcrowded, I am transferring a portion of this liver cell to another culture media, another vessel, same culture media, to another vessel containing fresh culture media. So this we can represent passage 1. Okay, and again it is subcultured. So how many number of times it is subcultured, that we can call it as the passage number. Okay, and population doubling level. So population doubling level, that is the specific number of cells involved, it is not at all relevant. Okay, so here the population doubling level depending upon the nutrients available each time in the fresh media, it can able to promote the population growth. Okay, here we can't able to correlate population doubling level with passage number. Let us say for example I am conducting the passage number is 4. The passage number 4 means 4 times I have conducted subculture. Here it does not mean here we can the population can be doubled 4 times, not like that. Okay, population doubling or population increase in population that takes place based on the amount of nutrients and other requirements available, other requirements how we are providing to the animal cells. Okay, it simply gives an indication how old the cells are. Okay, like how we are celebrating our birthday. Okay, the percentage number represents the age of the cell. So secondary cell culture and cell line. So primary cell culture is subcultured. Primary culture is subcultured. Then it becomes a secondary culture, or we can call it as a cell line or subclone. So here the process involves removing the growth media, and we are disassociating, that is separating the adherent cells by using enzymatic method. So subculturing of primary cells to different divisions, it will lead to generation of cell lines. Okay, by means of which we can able to generate the cell lines. So during the passage, the cells which is having the highest growth capacity, the cell which is having highest growth capacity, that results in the high degree of genotypic and phenotypic uniformity with the population. Okay, this genotype and phenotypes you will learn under your pharmaceutical biotechnology. in sixth semester. Separate topic is there, microbial genetics. Okay, so genotypic and phenotypic uniformity is there in the population, but they are subcultured serially. So there is a chances to become different from the original parent cell. Okay, so during the passage they are trying to carry the genotypic and phenotypic. It represents the morphological similarity. Okay, so morphological similarity or genetical similarity they try to maintain. But if we go for many number of subcultures, say for example if I have conducted 6 passages, okay, and while go for seventh subculture, if we compare the characteristic feature of cells which are grown in seventh subculture and the cells of primary cell subculture, there might be some deviations. Okay, so that is the thing that takes place with secondary cell culture. And here comes the other types of animal cell culture based on the lifespan. Already we discussed finite cell lines and continuous cell lines. So finite cell lines, so these cells grow through a limited number of division. So their lifespan is limited. So the cell passage several times, then they can able to lose their ability to proliferate. They could not able to reproduce or they could not able to multiply. Generally, the cells derived from primary cultures are finite cell lines. Continuous cell line, it undergoes transformation and it acquires the ability to divide indefinitely. So this transformation, how this transformation is nothing but change in the characteristic feature. Okay, so how this transformation come? Because of many number of passages. Because of many number of passages, they got transformed from their original characteristic feature. So they can able to divide indefinitely. So sometimes we can call this transformation as a type of mutation. And this mutation or transformation, it can occur spontaneously without any reason or without any mechanism. Just like that, it will take place spontaneously. And sometimes it can be done by using chemical method, or purposefully, artificially, we can induce the mutation to occur if we want to generate some needed tissues. So we can use some chemical or mechanical method, and we can make the cells to undergo indefinite cell division. Okay, so this can grow indefinitely as a permanent cell line, and they are immortal. Okay, these type of cells characteristic feature continuous cell lines, they are less adherent, fast growing and less fastidious in their nutritional requirements. So fastidious means they require some special nutritional elements to grow. Less fastidious, they are there is no conditional requirements. Okay, so if fundamental nutrients are available, by utilizing those it can able to grow, and it will be grow in the form of suspension, and it is having the tendency to grow on top of the each other, that we can call it as multi layers. Growth occur in multi layers on the culture vessel surfaces. Here it includes examples common cell lines. It includes human cell lines, that is MCF7 breast cancer, HL60 leukemia, then HELA human cervical cancer cells, and also primate cell lines. It includes Vero, that is African green monkey kidney epithelial cells. These are all few common cell lines that are developed from human beings and animals. So cell strain. So cell strain is nothing but the lineage of cell that is originated from the primary culture. Okay. So here there is a continuous lineage. Lineage is nothing but directly resulting from the ancestor. Okay. So directly resulting from the ancestor, that we can call it as the lineage. And these lineage of cells, it is also referred as the strain, or we can call them as the cell strain. So either derived from the primary culture, or they are derived from the cell line by positive selection or cloning techniques. Okay, they are having specific properties or characteristic feature, mostly which are similar to their parents or parental cells. So here a cell strain, it requires additional genetic changes subsequent to the initiation of parent cells. That is the important characteristic feature of cell strain. For the purpose of animal cell culture, the cells are taken from the organs of the experimental animal, and these cells may be removed directly or by means of mechanical method or by using some enzymatic action. Okay, so direct removal, mechanical method of removal, or enzymatic method of removal. So either of by these means we can able to remove the cells from the organs of experimental animal. So the cells which was obtained, it can be made as a cell line or we can call it as a cell strain. Okay, so which is nothing but again we are going deep into the cell. So thank you all for patience listening. And next comes the general method or procedure for preparing animal cell culture that you can see in my next presentation. So with this types, I'm concluding this presentation. Thank you all. We will meet again in next presentation. Thank you all once again.